Hi there. In this video, we're going to explore programs on Pluto with a focus on the translational alignment framework, and we're going to use this pediatric glioma resistance and immune state mapping program as an example. Pluto is an organizational intelligence layer for translational science, and it uses the translational alignment framework to automatically align all of your analysis and discovery work towards program language. So the highest level of organization on Pluto is programs, and you can use this table style view to see all of the details related to each program, and then use the Kanban style view to see where those programs sit at a glance and to move them between phases. On each program tile, there is a coverage plot showing how much evidence has been generated to support that program, and this coverage aligns to the Translational Alignment Framework, or the TAF, as I'll call it from here on out. So navigating into this program and going over to the framework, you'll see this radar plot that consists of 42 little wedges. And these wedges are criteria that are designed to help support translational program success. These criteria span early biology and disease discovery, target and therapeutic actionability, model and disease alignment, and mechanistic and functional confidence. And they have been defined by Pluto, but they can be configured specifically for your organization's science. You can see how each of these criteria are broken down by dimension. So for example, disease transcriptional signal is one of the criteria. We need to know the molecular changes distinguishing disease from control and generate an initial list of candidate genes involved in disease biology. So then you can see all of the plots or all the analyses that have been run to support that specific criteria, and then the rationale behind that specific plot. Based on how these criteria are filled out, Pluto automatically updates and shows the next steps and recommends what you should run next, or what analysis you should do next. Ultimately, all of these criteria are completed by uploading data and running analyses in Pluto. So every program contains an experimental dataset registry where you can import datasets from S3 or GCS, or just upload from your local device. Or you can also import from a global data commons. So these are the datasets that you want accessible across your entire organization. And then Pluto also supplies published datasets like TCGA and CCLE for you to utilize. Pluto supports a wide array of data types, from transcriptomics data types like RNA-seq, single-cell, and spatial, to epigenetics like ChIP-seq, ATAC-seq, and cut-and-run, to proteomics and metabolomics. For single-cell specifically, we'll just dive into this example. You can always upload raw FASTQ files or process data to Pluto, and then run that data through alignment and quantification pipelines, and these can be pulled from Pluto's repository of NF-core pipelines, or you can run your own in-house pipelines. And then for single-cell and spatial datasets, you can run additional annotation pipelines. So for example, you can do cell type annotation with CellKB or Gemini. Once a dataset has been processed, you can move on to analysis. So in Pluto, analyses are run in what we call a canvas or an analysis space, and typically an analysis space maps to one hypothesis or one scientific question, but you can use many different datasets to answer that question. So you can pull in datasets that are single-cell or RNA-seq or epigenetics, and we know that often the best targets are the ones where you see consistent patterns across omics modalities. So this canvas allows you to do that. You can run analyses using non-coding graphical modules and generate insights from them, and then you can even integrate this data across datasets with things like concordance analyses to identify those strongest targets. Once you've run analyses, you can then use this Align to Program tool to automatically align all of these plots, these analyses, back to the Translational Alignment Framework. So it will do the scoring and the categorization for you, and it will do it across all of your different analysis spaces with all of your different hypotheses and goals and questions. One thing that's important to note is that these analyses can be run manually by scientists and are designed to be very user-friendly for that reason. So they don't require any coding knowledge to generate these visualizations. However, they can also be run with agents. So you can define a hypothesis or question and then have Pluto automatically generate visualizations to answer that question. And similarly, with the Translational Alignment Framework, you can automatically align all of your data to specific criteria, but you can also do that manually and put your own manual rationale. Hopefully this gives you an idea of how Pluto allows scientists to analyze multiomics data independently, aggregate it, map it to program decisions, and make it visible to everyone on your team. Thank you so much for watching. Bye.